So I want to thank the organizers for inviting me here to present a pilot project for GP Write from our lab. So I'm Neville Sanjana. I'm here both at the New York Genome Center and a little bit uptown at NYU in biology, ISG, and neuroscience. And I'm going to tell you a little bit about work that we've been doing in the lab for a few years now to use CRISPR technology to do pooled screens, which is actually it's nice. The previous talk was a really nice introduction to the work that we're doing. So one thing that's been kind of a motivating factor for a lot of the work we've been doing has been the tremendous advances in genome sequencing. And this is echoing a slide shown earlier today by George. And this is just data, a little bit different data from the GWAS catalog from NIH and EBI, and just showing this tremendous increase in growth in the number of publications where we've taken groups of patients who have certain diseases, sequenced their genomes, and tried to find what genetic variants are different about those folks than folks who maybe don't have this disease. And so there's just been this tremendous growth in our ability to read out genomes, and as I think most of the people in this room are motivated by, it's not been mirrored by a similar ability to write genomes. And our ability to actually not even just write out a whole genome, but just to interrogate and perturb even specific variants is not really there at the same scale at which we can easily sequence. You know, all of you should have your genome sequenced by now, everybody in this room. And so we and many other labs have been very interested in using these molecular scissors called CRISPR for DNA that can be used to cut or to modify DNA. And often you might hear this colloquially referred to as cut and paste, just like you have cut and paste in Microsoft Word, cut and paste for the genome. But really, we've delivered well on the cut side of things and not as well on the paste side. And both of those are pretty important. So I'm going to give you a tiny bit of background on cut and then tell you about our pilot project, which is about paste. And so here with this Cas9 enzyme, which is the most, I guess, widely used of the CRISPR enzymes, it's targeted to a specific location in the genome by a small piece of RNA that guides it. And if it's targeted to a gene, it can cut this gene, and that can result in gene knockout very efficiently, as I'll show you in one slide. And if it's targeted to some non-coding region, we've heard a lot about the importance of regulatory variants in non-coding regions. That can also result in some mutagenesis there. So we can try and figure out what is that non-coding region, like the introns we just heard about, what does that do? So I and others have really capitalized on this idea that we can program the CRISPR system with these short RNA guides and taken advantage of oligonucleotide array synthesis technology, which I'm sure we're going to hear a lot about during this conference. And this thing can actually synthesize tens of thousands, up to millions of these guide RNAs. We can target many locations in the genome. And so now, a few years ago, the work that we did is we synthesized CRISPR guide RNA libraries that really can target every one of those 20,000 genes in the human genome. And this way we can test, by manipulating these genes, we can say which genes are involved in a neurodevelopmental disorder, which genes are involved in cancer drug resistance. That was one of the first things we actually did with this. And we can take advantage of the fact that it's very efficient using the CRISPR system to create these loss-of-function mutations. I just show you a tiny bit of data from this paper to prove to you how efficient it is. So this is a human cell line, HEX cells, a very common cell line, and that's just shown here in gray. This is fluorescence, GFP fluorescence. And in those HEX cells, we put in one copy of GFP, this green fluorescent protein from jellyfish. And so that makes the cells more green, more fluorescent. And what I'm showing you here is a few different CRISPR guide RNAs targeting this copy of GFP. So we introduced this gene in, and now we're trying to get rid of it. And you can see that basically 7 days after we introduced these CRISPR guide RNAs, they're basically become like the non-fluorescent parental cell line again. The GFP fluorescence is obliterated. And compared to something, a technology that's more that's been used for a longer time than CRISPR, RNA interference or knockdown, here we're seeing about 90% knockdown of the GFP, but this is just fundamentally different. This is complete knockout. So it's very efficient. We go and we sequence this GFP gene in these human cells, complete knockout, very, very efficient. But the problem, and this is a problem we've known about for some time, is that even though that knockout is quite efficient, and I should say this is not my data, this is data from the lab I came from Feng Zhang's group, knockout is quite efficient, that the efficiency of precise gene editing for introducing particular mutations, like a mutation we might find that's different between my genome and your genome, this is just a lower efficiency thing. And so this is the motivating factor of motivating result behind our pilot project. And that pilot project is developing high efficiency knock-in pooled screens. So just like we can go to many genes in the genome and knock them out, can we do the same thing with knock-in? And there's a few engineering challenges we need to overcome. One is we need to be able to put this into a viral delivery format, because there we can make sure that each cell only sees one manipulation. We need to be able to select for the construct itself. So this is a little bit of a technical detail, but it's something we discovered when doing knockout, is that if we can select for cells that actually got the CRISPR reagents, we see much, much higher levels of knockout compared to when we don't select. Also, can we optimize aspects of the DNA donor design to boost the efficiency of either homologous or non-homology-driven insertion methods? Single-stranded DNA, double-stranded DNA, asymmetrical, symmetrical. There's a lot of things that we can just play around with to see how do we boost this efficiency. And the other thing is, I told you we took advantage of oligonucleotide array synthesis to make tons of these CRISPR guide RNAs. Now we have a different problem. We have to pair the synthesis of these guide RNAs with actually a donor piece of DNA that includes a mutation that we're trying to introduce. So it's a small, each one is a small technical thing, but we have to address each of these if we want to be successful in this endeavor. And just to motivate you for why I think this is so important, just take an example from cancer genomics, something that's near and dear to my heart. This is a paper that was published last week in Nature, a really landmark paper, 200 tumors, whole genome sequencing of these melanoma tumors. Melanoma is a tremendously mutagenic cancer. It's caused by UV radiation in many cases, lots of mutations. And what these folks found in this work is that in each tumor they found an average of 100,000 single nucleotide variants, precise single nucleotide changes. And they found one tumor with almost 1 million of these. And how do we make sense of all these different variants rapidly? How do we know which ones are functional, which ones are drivers, and which ones are not? So many of these occur in coding regions, many occur in non-coding regulatory regions where we know even less. And the question we want to answer is, do these mutations just disrupt an existing functional element, or do specific mutations create new functions? And that's why we need the precise editing that we don't have with the work that we've done before. And I think this will have wide applicability over many areas. This is what we've done with the knockout screens. We've looked at drug resistance to therapies in melanoma, both small molecule therapies and immunotherapies. We've looked at tumor metastasis. How do tumors, what mutations enable tumors to go from one organ to another? And we've looked at it in non-cancer contexts like hemoglobin diseases and metabolic disorders. So with that, I'm being told I should definitely stop. So some people who funded our work, I'm very grateful for that funding, and the people who've joined me in the new lab here, and some collaborators from all around. And we are recruiting postdocs and PhD students, so feel free to contact me if any of this sounds interesting, and join us for our GP Write pilot.
I'm thinking about this, pairing the synthesis of the guide RNAs and the donor templates. I'm wondering whether this would have to go all the way to pairing it in the context of the complexes that go to the specific sites in the genome to have more efficient replacement of the actual nucleotides there. Do you think?
So far nobody's done that, but I think that's actually a really cool idea. So you can imagine having some DNA binding, in addition to the donor, you could kind of grab onto it some way with a DNA binding protein. Mm-hmm. And people have shown so many cool ways to tether things to Cas9, either with direct fusions or by introducing into the guide RNA. I showed you that one picture with that RNA molecule. So into those stem loops, people have added this is RNA binding protein, an MS2 protein, but you can imagine also having a DNA binding protein with like a specific DNA aptamer.
Yeah. Okay.
That's a great idea, actually. Yeah.
I have a quick question here. How do you overcome the PAM dependence of the CRISPR technology?
So that's a great question. There's been some nice work. There's some a very nice paper in Nature Genetics from a few months ago from Dan Bauer and Stu Orkin's group up at Harvard. And there they actually used multiple Cas9 orthologs that have different PAMs. So they, in that paper, they mixed S. pyogenes Cas9 with the NGG PAM with Staph aureus Cas9 with the NNRGGT PAM, I think. And so that increases. So Cas9 itself, I mean, people are worried about the PAM. There's 2 Gs in a row pretty much every 10 bases in the human genome. So it's not a huge issue, but you know, more PAMs increase the targetable space. There's no doubt. And okay, great. Thank you.